Good morning, dear students. Today we will be doing the chapter Absorption by Roots, the processes involved. So we will study why roots are best suited for absorption of water and nutrients from the soil. So let's begin. So first of all, you need to know why water is required in a plant. The water is required because it is a constituent of protoplasm. Every cell has a particular concentration, and for that, it requires water. It is required as one of the reactants in photosynthesis. Photosynthesis is a process by which green plants manufacture their food using carbon dioxide and water in the presence of sunlight and chlorophyll to produce glucose and give out oxygen. It is lost in transpiration, so plants need to compensate for that water. The moment the stomata open for exchange of gases during photosynthesis, water is lost alongside. So to compensate for that water, a transpiration pull is generated, which will draw in more water from the soil. It helps in transportation of substances, both minerals and prepared food. The minerals are transported upward through xylem, and the prepared food is transported downward through the phloem, either to the plant parts for storage or for usage. Then it provides a mechanical stiffness. If a plant is not watered, it will wilt and ultimately die. If, on the other hand, you water it properly, the plant parts will stand erect, and this is especially important in case of herbaceous plants, where they have no hard or woody structure to provide any mechanical stiffness. Now we will be studying why roots are best suited to absorb water. This was studied by a botanist called H.J. Dittmer, who worked out on a 4-month-old rye plant. He said that the aggregate length of entire root was about 600 kilometers. Imagine this is just one 4-month-old rye plant. The number of root here over 14 billion, and the estimated total length exceeded 10,000 kilometers. So this tells us why the root is best suited to absorb water. It has an enormous surface area. No other plant part can match this surface area. So the roots are best suited because they have enormous surface area, as can be seen by the small seedling. It has extensive root system. The size of the seedling and see the extensive root system. They have a cell sap at a higher concentration than surrounding water. They have thin walls, only a cell wall and cell membrane, and they have a large central vacuole for absorption of water. The water needs to be retained in the plant cell. It is retained in the vacuole. So this diagram here shows you the development stages of the root hair. The root hair is basically an extension of the single epidermal cell. Here it begins as a small outgrowth, which enlarges and ultimately develops to the full-grown size. It has a central vacuole filled with cell sap, which will be having large concentration of solutes to allow for easy access of water from outside. It has 2 layers surrounding it, a cell wall and cell membrane. So this makes the roots the best suited structures to absorb water. So once they are surrounded with soil water, the water readily passes in through a process which we will be studying later. That process is called osmosis. Now next, you are going to study the transport of substances across a cell, which occurs by 2 processes. Number 1, the passive transport. Number 2, the active transport. So passive transport, as the name indicates, is a type of transport which does not involve the usement of any energy in the form of ATP. It can be of 3 different types: diffusion, osmosis, and imbibition. And the active transport is the type of transfer which uses energy. So active transport can be from a region of low concentration to a region of high concentration, whereas the passive transport will be from a region of high concentration to a region of low concentration, with or without the aid of protein molecules. So let's study the passive transport now. So passive transport, we say, is the free movement of molecules from a region of their higher concentration to the region of their lower concentration without any input of energy. So this is of three types. The first one is called imbibition. Now in imbibition, the water is absorbed on the surface of protein or cellulose-based plant parts. In diffusion, the concentration gradient is from high to low. So the substances always move from their high concentration gradient to their low concentration. Osmosis is also from high concentration to low concentration, but this is restricted to a solvent, and generally the solvent happens to be water. There is one catch here. It is always across a semi-permeable membrane. So it is also diffusion of substances, the solvent, from a high concentration to a low concentration, but across a semi-permeable membrane. So let's study imbibition in detail now. Imbibition is the first process of passive transport. If you soak seeds in water overnight, bean seeds or any other plant seeds, you notice that they swell up in the morning. The cotyledons absorb water. The seed coat also ruptures in certain cases. And this phenomena takes place because these are either dead or living plant cells, and they have absorbed water by surface attraction. This is called imbibition. Okay, this process is called imbibition. The next you are seeing is a process, a type of imbibition noticed in case of wooden doors and windows. In the rainy season, they swell up, due to which they are not able to latch properly or close properly. They become uneven. This is because the wood is made up of cellulose, and cellulose also shows imbibition. So here the kind of imbibition displayed is called as warping. This kind of imbibition is found in wooden doors and windows because cellulose, it's a dead part of a dead plant cell that has swollen up due to imbibition. The next passive method you are going to study is called diffusion. It is a free movement of molecules of a substance. The substance can be solute, solvent, gas, liquid, from the region of their higher concentration to the region of their lower concentration when the two are in direct contact. Now, if you look at this gas jar, here you have potassium permanganate in its higher concentration. Here the concentration of potassium permanganate is less. So potassium permanganate diffuses upwards. But water concentration is high here, water concentration is low here. So water diffuses downwards till the time both of them have acquired a uniform concentration. This is called as diffusion. The next process is called as osmosis. Osmosis is movement of water molecules from the region of their higher concentration to the region of their lower concentration through a semi-permeable membrane. When you talk about higher concentration, you're talking about water molecules. Where the concentration of water will be higher, that solution will be a dilute solution. And where the concentration of water molecules will be lower, that solution will be a concentrated solution. The solutions, we will be studying the term tonicity later, but at present you need to know the solutions can be of three types. If you compare the solution inside the cell sap and the solution in which a plant cell is placed, they have same concentration. It is isotonic. In an isotonic solution, the number of water molecules entering inside the cell will be equal to the number of water molecules leaving the cell. The second is called as hypotonic. That means there is more water outside, less water inside the cell, due to which water tries to enter in the cell. The cell becomes turgid. The third one is called as hypertonic, wherein a cell which has more water as compared to the solution in which it is placed. Due to this, the water will leave the cell. The cell will become plasmolized. Now, before we proceed further with osmosis, there are these four types of membranes that you need to study in detail. The four types of membranes are impermeable, freely permeable, semi-permeable and selectively permeable or differentially permeable. So first we have impermeable membranes, the membrane which does not allow anything to pass through it. Anything in solution or otherwise will not be able to pass through this kind of a membrane. For example, rubber sheet. If you place anything inside a rubber balloon, nothing will pass through that rubber balloon because it is completely impermeable. Freely permeable, for example, muslin cloth or a cell wall. The plant cell wall will allow anything in solution to pass through. It will still not allow soil, rock, particles to pass through, but anything in solution will be allowed free movement. Then comes the semi-permeable membrane. For example, the egg membrane, the whisking bag, the goat's bladder, the sheep's bladder, the cellophane paper. They will allow solvent particles, small sized solvent particles to pass through, but their pores will not permit the bigger solute molecules to move across. Then comes the last one, selectively permeable or differentially permeable membrane. Now, selectively permeable membrane or differentially permeable membrane basically permits the movement of solvent molecules, that is water molecules, but if required in certain circumstances, it can permit the movement of solute particles also, but by involvement of energy in the form of ATP. That means through active transport, the solute molecules can move across the cell membrane. Therefore, cell membrane, the living membrane, is the only example of selectively permeable or differentially permeable membrane. Now, here comes the term tonicity, which I have been referring to in the previous few slides. So what is tonicity? It is the relative concentration of solutions, always two solutions compared together, which determines the direction and extent of diffusion. In what direction will the diffusion occur and to what extent will it occur? So it is of three types: isotonic, hypotonic, and hypertonic. Isotonic means the two solutions have the same solute solvent concentration. Equal amount of water molecules will move across the semi-permeable membrane. There is no osmosis in such a case. Hypotonic, when the solution outside the cell has lesser solute and inside has more solute, due to which endosmosis, that is inward movement of water, will take place in a cell. Hypertonic, that means the solution outside the cell has more solute than the solution inside the cell sap, due to which water will be drawn out of the body of the cell, which is called as X-osmosis. Now, whenever we talk about movement of water across a semi-permeable membrane, you consider the two different solutions together. So always water moves from a dilute to a concentrated solution. Dilute to a concentrated would cause a rise in the level of water in one side. So here from A to B, the water is moving because water is more in A than in B. And due to this, there is a difference in the water columns on the two sides. After some time, the process of osmosis stops. No further rise in water will take place. Water is basically trying to equate the concentration on both sides. But the concentration can be equated only in terms of water. So water tries to move. After some time, this solution will prevent entry of any more water and the process of osmosis will stop. So if you see it here, you have two comparison of diffusion and osmosis. So in diffusion, the salt and the water will mix freely. There is no membrane separating them. But in osmosis, there is a semi-permeable membrane separating the two different solutions having two different tonicities, due to which there is a difference in the water column. One side will show endosmosis, another side will show X-osmosis. So if you see this part, A is showing X-osmosis, B is showing endosmosis. They will try to equate the concentration of water on both sides. Okay, and this can be demonstrated by the Thistle funnel experiment. So this is how the Thistle funnel is set up in the lab. You place a concentrated solution, you mark the initial level, in the concentrated solution, you place it in a beaker full of water and you leave it for some hours. After some hours, the rise in level takes place from A initial to B final, which will tell you end osmosis has taken place across this semi-permeable membrane. Okay, so this type of osmosis is called as end osmosis, when there is a rise in the water column or the solution level inside the Thistle funnel. This is because a semi-permeable membrane will permit the entry of water molecules from water to the sugar solution inside. Because sugar solution is hypertonic, water is in comparison hypotonic. So water concentration is high outside than inside. So water will go from outside to inside. The water outside will not become sweet if you taste it after some time, indicating that only the movement of water has taken place inside. The sugar has not come out of this semi-permeable membrane. Or in other words, we also understand that the semi-permeable membrane is not permeable to sugar.